Greetings to everyone. Welcome back once again to this educational channel called Learn Bio with Janet. I'm teacher Janet, and today we will be discussing Chapter 11 of the Form 4 Biology syllabus, KSSM syllabus. We'll be studying 11.3, subtopic 11.3, types of immunity. The learning outcomes for today's lesson are as follows. For subtopic 11.3, types of immunity, after this lesson you should be able to communicate about the types of immunity, that is A, active immunity, and B, passive immunity. For active immunity, there are two types, natural active immunity and artificial active immunity. And similarly, for passive immunity, there are two types, natural passive immunity and artificial passive immunity. So we have to understand and also be able to explain these types of immunity. First, let us define immunity again. Now, immunity is the ability of an organism to resist infection or disease and to defend itself or himself or herself against infection by pathogens. So, immunity is the ability of an organism to resist infection. Resist means to ward off the infection so that it will not cause sickness. It will not cause sickness in the body. Alright? And to defend itself or oneself against infection by pathogens. That means the body is able to defend itself by destroying the pathogens that have infected the body. How does it defend itself? By producing specific antibodies that promote the destruction of the pathogens. In that way, the body is once again free of the pathogens and the person will not fall sick. Right? So, infection here means invasion of the body by pathogens or disease-causing microorganisms. So, we live in a world where there are microorganisms or disease-causing microorganisms everywhere. And these pathogens or disease-causing microorganisms are always trying to find ways to enter the body because they are parasites and they need to live in the living host and to get their nutrition from there and also to reproduce. So the human body has to constantly fight off these pathogens. And if they were to invade the body, then the body's lymphocytes must then take action and produce antibodies to destroy the pathogens or to promote destruction of the pathogens. Okay? So we are in a sort of cell war with the pathogens all the time, right? So immunity is the ability of an organism to resist infection or disease and to defend itself against infection by pathogens. So if you have immunity against a disease, it means that you're immune to the disease and you will not fall sick even if the pathogen tries to invade your body. You can defend yourself and then destroy the pathogens. Okay? So immunity depends on the presence of lymphocytes which produce specific antibodies during infection. And this is called a specific immune response. So how does immunity come about? It depends on the presence of lymphocytes in our body. These cells, they have the round nucleus, big round nucleus, and they are white blood cells or leukocytes, special white blood cells, which can produce specific antibodies to destroy specific pathogens. All right? So the antibodies are specific for each type of pathogen, meaning that one type of antibody can only act on and destroy one type of pathogen. Now, immunity can be divided into two types, active immunity and passive immunity. In active immunity, the body's own lymphocytes produce their own antibodies when they are stimulated by antigens, which are foreign particles that have entered the body, such as pathogens that have entered the body. So these antigens will stimulate the body's own lymphocytes to produce specific antibodies to destroy the antigens. Okay, so the body was active in producing its own antibodies. So that's why we call it active immunity. So active immunity can be divided into natural active immunity and artificial active immunity. In natural active immunity, the person falls sick, recovers, and then when he falls sick, he produces the antibodies against the disease, and then when he recovers, he has a high level of the antibodies already. So that means he has natural active immunity. For artificial active immunity, the word artificial here means that it usually involves an injection. All right. So in artificial active immunity, a vaccine was injected into a person in order to stimulate the lymphocytes to produce their own antibodies. We'll discuss all this later. Okay. So in active immunity, pathogens stimulate lymphocytes to produce their own antibodies in the person's body. Now, what about passive immunity? Passive immunity is obtained when the body does not produce the antibodies itself, but it receives the antibodies from an external source, such as from the mother. The baby receives the antibodies from the mother by from breast milk. Okay, and then for another way is when the antibodies are extracted from an animal's blood and then injected into a person. Okay, it means the antibodies come from the animal, not from the person himself. So that is passive immunity because the body was passive. It did not actively produce its own antibodies, but received the antibodies from an external source. Okay, so passive immunity can be divided into natural passive immunity and artificial passive immunity. So in natural passive immunity, the word natural here means that it was acquired through natural means, such as when the mother breastfed the baby. The baby will receive the maternal antibodies from the mother, right? And for artificial passive immunity, the person acquires this type of immunity through the injection of a substance called antiserum, which are actually ready-made antibodies from an animal's blood, usually from an animal's blood. Okay, right. Now, both active and passive immunity can be naturally or artificially acquired, as we have discussed briefly here. So here we are going to look at a very useful overview of the different types of immunity. So immunity can be divided into two basic types: active immunity and passive immunity. In active immunity, a person's own lymphocytes produces his own antibodies when stimulated by antigens. But in passive immunity, the body receives antibodies from an external source. Now, active immunity can be divided into natural active immunity and artificial active immunity. In natural active immunity, a person falls sick when infected, and then his lymphocytes start to produce specific antibodies in order to destroy the pathogens. So after that, he recovers from the infection and he will have a high level of that specific antibodies already. So he has gained natural active immunity. For example, for diseases like chickenpox and measles. For artificial active immunity, an injection is given. A vaccine is injected into the body to stimulate the body to produce its own antibodies, to stimulate the body's lymphocytes to produce antibodies against the vaccine's dead antigens. So a vaccine contains dead or weakened antigens, such as dead pathogens. Okay, and then it will give the lymphocytes a sample of these antigens that would stimulate them to produce specific antibodies against the specific antigen. So the person doesn't fall sick when he's given a vaccine, but he still managed to produce his own antibodies against that specific antigen. Now, for passive immunity, the body receives antibodies from external source. So it's divided into natural passive immunity and artificial passive immunity. Now, in natural passive immunity, the baby receives antibodies from the mother while breastfeeding, because the mother's breast milk will have antibodies from her, the maternal antibodies from her. Or during pregnancy, the fetus can receive maternal antibodies through the placenta, which is an organ that supplies the fetus with nutrients and other substances. So the antibodies can also diffuse from the placenta into the fetus. Now, for artificial passive immunity, an anti-serum which contains ready-made antibodies is injected into the body of a person, especially when the person is already suspected to have been infected by pathogens, let's say. Then he'll be given this anti-serum containing the antibody to destroy the pathogens that may have entered his body. So the anti-serum containing antibody actually comes from an animal, usually an animal that has been intentionally made sick or injected with these pathogens so that the animal's lymphocytes can produce the antibodies. Then the antibody is extracted and then injected into a person who may be sick from this from the disease caused by that specific pathogen. So now we are going to go into more details about the different types of immunity, and we start off with natural active immunity. Okay, so when someone is infected by a pathogen, that means the pathogen has invaded his body, his own lymphocytes will produce antibodies as a response to these antigens. So these antibodies are proteins or substances that can, they are substances that can bind to the antigens and then promote the destruction of the antigens in the body. Now, the level of these specific antibodies will gradually increase as the person is falling sick, from chickenpox, for example. So once the chickenpox virus has entered his body, his lymphocytes will start producing the antibodies, and the level of antibodies will gradually increase in the blood until it reaches a level which is high enough for him to be able to destroy all the pathogens. So that level of immunity must be reached. It's a level of antibodies that is needed for the person to attain immunity, for the person to destroy all the pathogens, okay, for that particular disease. So once his antibody level reaches the level of immunity, he has immunity already. So he recovers from the disease, and then he gains a permanent immunity against the disease. As we said, examples are like for chickenpox and measles. So the person can be said to be immune to the disease already after he has suffered from chickenpox for the first time, because he's immune because he has a high level of antibodies that can defend him against the pathogens, the same pathogens. Furthermore, he has memory cells. Okay, memory cells are actually lymphocytes that have stored memories of the pathogen and also of the antibody needed to destroy the pathogen. So when an individual is attacked again by the same pathogen, for example the chickenpox virus, he doesn't fall sick because the lymphocytes that stored the memories of that pathogen and that can produce the specific antibody, they are still around, some of them, from the first infection. So they are called memory cells. They are in the blood and they are ready, right? Because they can produce that specific antibody already. So they will rapidly produce the specific antibodies to react immediately against the antigens and to destroy them. So it will destroy the antigens. The antibodies will destroy the antigens before the person falls sick. So the person will not fall sick from chickenpox again, and he's considered to be immune. All right. So anytime the chickenpox virus tries to infect that same person, his memory cells are already ready to rapidly produce antibodies to destroy the chickenpox virus. Okay. Here's the graph for natural active immunity. For example, at the point here, pathogens infect the body at 0 weeks. And this stimulates the lymphocytes to start producing antibodies. So the concentration of antibodies will increase gradually with time as the lymphocytes produce more and more of these specific antibodies to destroy the pathogens. So, but then if the level of immunity is not reached, which is the concentration of antibodies needed to obtain immunity, then if this level of immunity is not reached yet, the person is not yet immune, and he falls sick from the disease because the pathogens are not totally destroyed and they attack the body. But as the level of antibodies increases and goes beyond the level of immunity, he's able to destroy all the pathogens in the body, and so he becomes immune, and then he recovers from the disease. All right? After that, you will have a high level of these antibodies, but the antibody level can decrease as time goes by. All right? So for the first infection, there is slower production of antibodies by the lymphocytes. Slow production, because the lymphocytes are just beginning to produce the specific antibodies for that pathogen. However, if there is another infection later on, after maybe a year or two or and so forth, during the second infection by the same pathogen, memory cells, which are lymphocytes that can produce these specific antibodies, will produce a more rapid immune response, meaning they will produce more of the same antibodies, and also they'll produce the antibodies faster, okay, than the first time. So the gradient for this second curve is steeper than for the first curve. And because they produce these antibodies faster, they are able to destroy the pathogens faster, and they will not fall sick because of the disease. Okay? So the level of antibodies will increase very fast and exceed the level of immunity. All right? For the second time. But this time it's faster. Production of antibodies is faster, and more antibodies will be produced. All right? So the person will not fall sick from the disease, and we say that he is immune to the disease. Now, the second type of immunity is called artificial active immunity. So it is obtained through immunization. What is immunization? It is the process that stimulates immunity against a specific disease through injections of vaccine. Okay, so injection of vaccines are sometimes known as, the process is known as vaccination. Okay, so immunization is acquired through vaccination. All right, so when you carry out immunization, you're actually trying to make a person immune towards a disease through the injection of vaccine. So the aim or the function of this immunization is to protect oneself from being infected by a highly infectious disease such as tuberculosis or chickenpox, or nowadays we have this COVID-19 disease caused by the COVID-19 virus. So what is a vaccine actually? Now, a vaccine is a suspension of pathogens, disease-causing microorganisms, or even toxins that have been weakened. They are dead, for example, dead pathogens, or made non-virulent. Okay? Non-virulent means made it so that they cannot cause harm to the body, cannot cause disease anymore. So when a vaccine is injected into the body, as seen here, the vaccine will stimulate the body's lymphocytes to produce specific antibodies to fight the pathogen, the weakened pathogen, without the person falling sick. Now, the first vaccine injection may result in a low level of antibody production. That means the production of low levels of antibodies only. Maybe because there's not enough stimulation yet. And this may not be sufficient to protect an individual from the disease because the low level of antibodies has not reached the level of immunity. It's too low. Okay, so a booster dose may be given to increase the antibody production by the lymphocytes. In order for the level of antibodies to reach the level of immunity, then the person becomes immune to the disease and his level of antibodies is high enough so that they can destroy the pathogens should they enter the body. Can destroy all of them quickly too, and that will protect the individual from the disease, even though the pathogens try to infect the individual. They'll all be destroyed by the antibodies, which are at a high level. So after a person has been injected with a vaccine for a particular disease, he will have a high concentration of antibodies for that disease, and also he will have memory cells that have the memory of that particular pathogen and are able to produce the specific antibodies against that pathogen. So now, if he's infected by the actual pathogens, his memory cells or lymphocytes will be able to produce enough antibodies in large amounts and very rapidly to immediately destroy these pathogens. So he will not fall sick. And we say that he has become immune to the disease. Now, examples of vaccines are Salk vaccine for poliomyelitis. Poliomyelitis is a disease that causes paralysis because the virus attacks the nerves and the spinal cord. In Malay, we call it penyakit lumpuh. Then there is the BCG vaccine for tuberculosis or penyakit batuk kering. In tuberculosis, the lungs are attacked by bacteria. HPV vaccine is for cervical cancer, to prevent cervical cancer in women. Hepatitis B vaccine is to prevent hepatitis B, which is a liver disease. Rubella vaccine is given to women to prevent the disease called rubella. Now, actually, rubella disease is a non-fatal disease, but it is very dangerous to pregnant women who have fetus because the rubella virus attacks the tissues, the young tissues of the fetus in a pregnant woman, and can cause heart defects and also other serious effects. Okay, so that's why it's advisable for women to be given the rubella vaccine so that they will be able to resist the rubella virus, and in this way the fetus is protected should the woman become pregnant. Now, the word BCG vaccine for tuberculosis stands for Bacille Calmette-Guerin. So this is a vaccine. This graph shows us how the concentration of antibodies increases or changes with time for artificially acquired active immunity when injection of vaccine is given. So let's say we start from this point here at 0 weeks. Let's say the doctor has given the first injection of vaccine to an individual. So the vaccine contains the weak or dead pathogens or even toxins of disease. So when it is injected into the person, it stimulates the lymphocytes to produce antibodies to destroy the pathogens or to neutralize the toxins. All right. So the lymphocytes produce antibodies to destroy these antigens. Therefore, the level of antibodies start to increase. However, if the stimulation is not enough, after a while, the production of antibodies will stop and then the level of antibodies will drop down again. So maybe after the first injection, the antibody level is still below the level of immunity. All right, which is this level of immunity is the level of or concentration of antibodies that is needed for the person to become immune or resistant to the disease. So therefore, the person is still not immune to the disease at this point here. Thus, a second injection of vaccine may be needed, and this is called the booster dose. Sometimes there's even another booster dose, a third injection of vaccine. But anyway, in this case, in the second injection of vaccine or the booster dose, some more antigens, some more weakened pathogens or toxins are injected into the person. And now this time, when the lymphocytes are exposed to the same antigens again, the second time, the production of antibodies is now faster and also greater than the first time here. All right, you can see the curve here is steeper than the curve for the first injection. This shows that the production of antibodies in the second injection, after the second injection, is faster. The production of antibodies is faster, and also more antibodies are produced. And the level of or concentration of antibodies has exceeded the level of immunity, so the person has become immune at this point here, and is able to defend himself against any attack by the pathogens in future. Okay, so the graph for artificially acquired active immunity is like two mountains, okay, a lower one with a lower peak and a higher mountain with a higher peak. We must also know when the first and second injection of vaccine was given. We must be able to indicate it on a graph. Now for the second injection of vaccine, it is the point where the level of antibodies has dropped down and then it starts to increase again. That is the point where the second injection of vaccine was given. Now a common question asked in the exam is, why is a second dose or booster dose of vaccine needed? And here's the answer. Now the second dose or booster dose is needed here, okay, to further stimulate the lymphocytes so that they will produce more antibodies in order to increase the concentration of antibodies so that it exceeds the level of immunity. And once the concentration of antibodies exceeds the level of immunity here, the person has become immune to the disease and he can resist any infections by the pathogen if it occurs in the future. So he has acquired artificial active immunity. Okay, and another point to note is that the first injection did stimulate the production of antibodies, but not to the level of immunity. So the first injection is not enough to make the person immune to the disease. That's why the second dose is given to further stimulate the lymphocytes so that the production of antibodies will increase and also the concentration of antibodies will exceed the level of immunity. That's all for this lesson. In the next lesson, we will continue to study types of immunity. That will be part 2, and we'll discuss passive immunity in more detail. That is, natural passive immunity and artificial passive immunity. So thanks for watching, and do remember to share, like, subscribe, and comment. Goodbye for now.